Hello everyone. Today I would like to talk about differential accumulation of potassium in the vegetative and reproductive organs of 3 grapevine cultivars. We all know that potassium is an essential element for all living organisms. In plants, potassium is involved in numerous biochemical and physiological functions, including photosynthesis, maintenance of cell membrane potential, enzyme activation, cellular membrane transport, assimilate translocation, control of plant-water relations, turgor maintenance, and transpiration. In grapevines, just like any other plants, potassium deficiency can cause issues like reduced vine growth, premature leaf drop, and yield loss. Potassium deficiency symptoms in the leaf can occur as scorching and necrosis of leaves, starting from the margins of the leaves and advancing towards the center of the leaves. You can see in this picture, this is a potassium deficiency symptom seen on leaves in grapevines. When we consider grapeberries are a strong sink for potassium. In grapeberries, potassium accumulation occurs throughout the growth and development, with a slow potassium accumulation phase from fruit set to verizon and a rapid potassium accumulation between verizon to maturity. When we consider individual berries, a large portion of potassium goes to skin compared to the other components. Seeds and pericarp also accumulate some potassium, but a lesser proportion compared to the skin. When compared to the other cations like calcium and magnesium, potassium is the most abundant cation in grapeberries. Berry potassium is very important for determining berry color and berry quality. Even though potassium is such an important element for grapevines and grapeberries, high berry potassium can be an issue in wine industry because berries with the really high potassium will be associated with the high grape juice pH. And in winemaking, this can lead to wines with a low acidity with a flat taste. Wines with a high pH often subject to oxidative and biological spoilage. And high pH in wine can cause color instability and quality issues in wines. I'm sure most of you would be aware of this issue in the wine industry. Not only in Australia, this is seen in many wine growing regions all over the world, especially in warm climatic regions. Managing potassium in the vineyard is tricky because we need to ensure adequate potassium status to the vines for the growth and physiological functions. At the same time, we need to avoid excessive berry potassium accumulation. Current recommended practices for assessing vineyard nutritional wine potassium status is soil analysis and leaf analysis. Soil analysis has some limitations because usually soil potassium levels not correlate with the tissue potassium levels. Today I'm talking more about leaf analysis. For leaf analysis, 2 time points are recommended. Petiole analysis are the most common practice. It is recommended to collect petioles opposite to the basal punch for analysis. This table shows the grapevine leaf analysis standards used in Australia. It shows the concentration of potassium as a percentage of dry weight in these tissues. And this is the recommended adequate range of potassium concentration in these tissues. Here, what we do not know is whether this potassium concentration in these vegetative tissues are a good indicator of berry potassium. The relationship between berry potassium, leaf potassium, leaf laminae potassium, and leaf petiole potassium is not clear. This information is really important to draw meaningful conclusions from the tissue test results to predict berry potassium or manipulate berry potassium in the vineyards. So the objective of this study was to assess current practice of leaf analysis for assessing berry potassium. More specifically, to compare leaf tissue potassium with the reproductive tissue potassium in on-rooted Cabernet Sauvignon, Shiraz, and Chardonnay wines at flowering and verizon, all wines grown under same soil and management conditions. Also, the objective was to describe the interaction of berry potassium accumulation with the berry weight and berry potassium accumulation interaction with the bunch weight for these three economically important Australian wine grape cultivars. This study was conducted at the National Wine and Grape Industry Centre at Charles Sturt University in Australia. And this was done in a replicated variety block. And the trial design was completely randomized block design. We selected Shiraz, Chardonnay, and Cabernet Sauvignon varieties for this trial. All these wines were planted 15 years ago and on roots. What we did was, at flowering, we collected the inflorescence and the opposite leaves from two node positions. The node positions, the lower node position was the basal bunch position, and the upper node position was the subsequent upper node. And we separated the leaf into petiole and laminae. From the inflorescence, we separated the flowers, and then we separated the peduncle between the shoot junction and the first lateral of the rachis. At verizon, exactly the same way, we collected the bunches and the opposite leaves from two node positions and separated the berries, also separated the bunch stem and leaf laminae and petiole. Then all these tissues were dried and ground and analyzed for potassium by ICP-OES. So here it comes the results. This graphs here shows the average berry dry weight, average berry fresh weight, and potassium content per berry in these three cultivars. There was significant difference between the cultivars in average berry dry weight and average berry fresh weight. Also, there was significant difference between the cultivars in potassium content per berry. And berry potassium concentration as a percentage of dry weight or as a percentage of fresh weight was not significantly different between these cultivars. Also, potassium content per bunch was not different between these cultivars. This graph here shows potassium concentration in different tissue types at flowering and verizon in three cultivars. There was significant difference in potassium concentration between the tissue types, also difference in potassium concentration between the cultivars for each tissue type, and there was significant difference in potassium concentration between the time points. In leaf laminae, the potassium concentration was the lowest in all these three cultivars at both time points compared to the other tissue types. In Cabernet Sauvignon and Shiraz at Verizon, bun stem potassium and petiole potassium was highest at Verizon, but Chardonnay was different from these two varieties in that when we looked into the correlations of potassium concentration between different tissue types, interestingly, only bun stem potassium and petiole potassium were correlated at both time points. Flower potassium or berry potassium was not correlated with any of the vegetative tissue potassium levels. So to make it simple, I'm presenting the node position comparisons separately in this table. And at flowering, there was some significant difference between the lower node and upper node for some of these tissues in all these varieties. But at Verizon, there was no significant difference between the node positions. So in summary, potassium concentrations in the vegetative parts, which are recommended in the current tissue analysis protocol, are not correlated with flower potassium or berry potassium concentrations. The lack of correlation suggests that potassium concentrations in these vegetative organs may not be a reliable indicator of berry potassium concentration. Significant differences in potassium levels between cultivars and between organs and between the phenological stages indicate the importance of cultivar-specific potassium standards for these different time points. So I would like to acknowledge the National Wine and Grape Industry Center, the Department of Primary Industries, the New South Wales Wine, Charles Sturt University, and especially Wine Australia for supporting this work. And thank you all.